Basics of Medical Biochemistry. First edition, 2022. Chapter 6. Chemistry of Enzymes, Part 2 of 2. By Professor Dr. Muhammad Jawad. MBBS, Tao, M. Philosophy, Ph.D., Dip M.E. Head of the Biochemistry Department-Fazaia Ruth Pfau Medical College-PAF Base Faisal-Karachi-Pakistan. Ex-Professor of Biochemistry, Qassim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. 6.6 Enzyme Kinetics. Enzyme kinetics is the study of the rates of chemical reactions that are catalyzed by enzymes. It is concerned with the investigation of how enzymes bind substrates and turn them into products. It provides information about the catalytic mechanism of the enzyme, its role in metabolism, how its activity is controlled in the cell, and how drugs and poisons can inhibit its activity. Initial work on enzyme kinetics was done by Victor Henry in 1902. Further deep studies were carried out by two scientists in 1909, German chemist Leonor Michaelis and Canadian researcher Maud Leonore-Menten. They proposed a quantitative theory of enzyme kinetics called Michaelis-Menten Kinetics. According to this theory, enzyme-catalyzed reactions occur in two stages. In the first stage, the substrate binds reversibly to the enzyme, forming the enzyme-substrate complex. This is sometimes called the Michaelis complex. The enzyme then catalyzes the chemical step in the reaction and releases the product. Michaelis and Menten proposed a simple model that explained most of the features of an enzyme-catalyzed reaction. It is represented by the following equation. K1 K3 E plus S E S E plus P K2 E equals concentration of total enzyme, free plus combined. ES equals concentration of enzyme substrate complex. ES equals concentration of free enzyme. S equals concentration of substrate. Michaelis and Menten suggested that K2 K1. Brigg and Halden suggested that K2 plus K3 K1. Forward reaction V1 equals K1E, ESS. Reversed reaction V2 equals K2 ES. At equilibrium, V1 equals V2. K1E, ESS equals K2 ES. Or K2E, ESS. K1 ES. ES, ESS. ES. ES ESS. ES ES. ESS. ES. Or ES. ES. ES, A. Km plus S. But initial velocity, VO, ES. VO equals K3 ES. B. Put the value of ES from EQ, A, into EQ, B. K3 ES, C. Km plus S. Maximum velocity is achieved when the maximum number of enzyme molecules is bound with the substrate. Vmax equals K3E or K3 equals. Put the value of K3 from above EQ in EQ, C. Vmax ES. EKM plus S. Vmax S. Km plus S. This is the Michaelis-Menten equation. Assumptions of the Michaelis-Menten equation. The concentration of substrate S is much greater than the concentration of enzyme E. ES does not change with time. It means that the formation of E plus S is equal to E plus P. Initial velocities are used in the analysis of enzyme reaction, first-order reaction, because maximum velocity is independent of the concentration of the substance, zero-order reaction. Lineweaverberg plot an equation. It is obtained by plotting 1/VO vs 1/S to get a straight line. It is also called a double reciprocal plot. Significance, it is used to calculate Km, calculate Vmax, determine the mechanism of enzyme inhibition. Figure 6.20 Lineweaverberg plot an equation, VO, initial velocity, Vmax, maximum velocity, Km, Michaelis-Menten constant, S, substrate concentration. The efficiency of an enzyme can be expressed in terms of kcat/kilometer. This is also called the specificity constant and incorporates the rate constants for all steps in the reaction. 6.7 Factors affecting enzyme action. The activity of the enzyme is affected by many factors like concentration of enzyme, the concentration of substrate, temperature, pH, presence of metal ions, coenzymes, and inhibitors. 6.7.1 Effect of enzyme concentration. The rate of reaction is directly proportional to enzyme concentration provided that the amount of substrate is constant. This is called pseudo-first-order enzyme catalyzed reaction. Figure 6.21 Effect of enzyme concentration on rate of reaction, E, concentration of enzyme. 6.7.2 Effect of substrate concentration. The effect of substrate concentration on enzyme kinetics, rate of reaction, is studied by plotting the rate of reaction, velocity, on the y-axis and the substrate concentration S on the x-axis. At low values of S, the VO rises almost linearly with increasing S. Any further increase in S does not affect the velocity of the reaction. Eventually, the enzyme becomes saturated with the substrate and the reaction attains its maximum velocity, Vmax. Michaelis-Menten constant, Km. The substrate concentration at which the rate of reaction is 1/2 Vmax is called the Michaelis-Menten constant, Km. Large Km. A large value of Km reflects a low affinity between enzyme and substrate. Small Km. A small value of Km reflects a high affinity between enzyme and substrate. Enzymes having small values of Km can achieve 1/2 Vmax at a low concentration of substrate. First-order reaction, the part of a reaction in which the rate of reaction is increased by an increase in substrate concentration. Zero-order reaction, the state of reaction when constant maximum velocity, Vmax, is achieved and no further increase in the rate of reaction is observed on increasing the substrate concentration. Figure 6.22 Effect of substrate concentration on rate of reaction. 6.7.3 Effect of temperature. Most body enzymes worked best at a normal body temperature of 37 degrees Celsius. A slight increase in temperature increases the rate of reaction but up to a certain limit because enzymes, being proteins, undergo denaturation at a higher temperature. Some plant enzymes act best at about 60 degrees Celsius. A few bacteria have enzymes that can withstand very high temperatures up to 100 degrees Celsius. However, most enzymes are fully denatured at 70 degrees Celsius. Optimum temperature. It is the temperature at which the activity of an enzyme is best. Most human enzymes have an optimum temperature between 35 degrees Celsius to 40 degrees Celsius. Their denaturation starts at temperatures above 40 degrees Celsius. The temperature coefficient, Q10, it is the increase in the rate of reaction for a 10 degrees Celsius rise in temperature. For chemical reactions, the Q10 value of 2 to 3 means the rate of the reaction doubles or triples for every 10 degrees Celsius rises in temperature. The optimum temperature for an enzyme-controlled reaction is a balance between the Q10 and denaturation. Figure 6.23 Effect of temperature on the rate of reaction. 6.7.4 Effect of pH. Different enzymes worked best at different pH, called optimum pH. For example, the optimum pH of pepsin is 1.5 to 2.0, for amylase, it is 6.4 to 6.9 and for alkaline phosphatase, it is about 9.5. The structure of the enzyme is changed in an environment with unacceptable pH levels. The extreme change in pH levels produces denaturation by distorting the active site of the enzyme in such a way that the substrate molecules will no longer fit in it. The pH values different from the enzyme's optimum value, cause changes in the charges of the enzyme and its substrate molecules. This change in ionization affects the binding of the substrate with the active site. Figure 6.24 Effect of pH on the rate of reaction. 6.7.5 Effect of product concentration. The rate of an enzyme-catalyzed reaction is also affected by the concentration of products. As most reactions of the body are reversible, that's why products formed at the end may start to become reactants, and thus the rate of reaction decreases. If products are removed continuously, then the rate of reaction will not slow down and the reactants may be completely converted to products. 6.7.6 Effect of the presence of metals. Some enzymes require metal ions as cofactors for activity. Metal-activated enzymes have an absolute requirement or are stimulated by metal ions like K+, Ca2+, and Mg2+. For example, Cl activates salivary amylase and Ca2+ activates the thrombokinase enzyme. Whereas metalloenzymes contain firmly bound metal ions, iron, zinc, copper, cobalt, at the active site of the enzymes. 6.8 Inhibition of enzymes. Enzyme inhibition refers to the interference in the working of the enzyme by another molecule. Inhibitors are chemicals that reduce the rate of enzyme-catalyzed reactions. They are usually specific and work at low concentrations. They block the enzyme but they do not usually destroy it. Inhibitor, I, binds to an enzyme and prevents the formation of ES complex as well as E plus P. Many drugs and poisons are inhibitors of enzymes in the nervous system. Enzyme inhibition can be reversible or irreversible. 6.8.1 Reversible enzyme inhibition. In reversible inhibition of the enzyme, the linkage between inhibitor and enzyme is non-covalent and it is usually by hydrogen bonds, electrostatic bonds, and van der Waals forces. This inhibition can easily be reversed by dilution, i.e., by adding more substrate. The enzyme is inactive only when an inhibitor is present. There are three basic types of reversible inhibition: competitive, non-competitive, and uncompetitive. These can be distinguished experimentally by their effects on the enzyme kinetic patterns. 6.8.1.1 Competitive inhibition. It is further divided into the following two types. I. Classical competitive inhibition. In this type of inhibition, there is a great structural similarity between the substrate, S, and inhibitor, I. S and I can bind to the same site on enzyme, E. The presence of I on E prevents the formation of ES complex. For example, malonic acid is a classical competitive inhibitor of succinate dehydrogenase. The antibacterial effect of an antibiotic is developed on this principle. Example: sulfonamides act by blocking the synthesis of folic acid. Sulfonamides are structural analogues of para-aminobenzoic acid, which is a part of folic acid. Sulfonamides act as competitive inhibitors of the enzyme dihydroptate synthase, DHPS, an enzyme involved in folate synthesis. Sulfonamides are therefore bacteriostatic and inhibit the growth and multiplication of bacteria. Table 6.6 Clinical use of some competitive inhibitors. Inhibitor. Antimetabolite. Enzyme inhibited. Clinical use. Sulfonamide. PABA synthetase. As antibiotic. As azrine. Glutamine oxidase. As anti-cancer drug. Prevent the synthesis of purine. Allopurinol. Xanthine oxidase. Treatment of gout. Inhibit the synthesis of uric acid. Acetazolamide. Carbonic anhydrase. As diuretic. 2. Non-classical competitive inhibition. In this type of competitive inhibition, the I binds to a site other than the active site and indirectly prevents the binding of S to E. But if S is already bound to the active site of E, then it prevents the binding of I at a separate site. Figure 6.25 Competitive inhibition of the enzyme. A. Classical. B. Non-classical. Effects of competitive inhibition on enzyme kinetics. IKM is increased. 2 Vmax remains unchanged. 3 lines on a double reciprocal plot intersect at the same point on the y-axis. 6.8.1.2 Non-competitive inhibition. In this type of reversible inhibition, the inhibitor binds to either enzyme or ES complex to inactivate the enzyme. There is no structural resemblance between I and S. The presence of I produces certain conformational changes in the active site of the enzyme in such a way that it will not accept substrate. Non-competitive inhibitors are not influenced by the concentration of the substrate, i.e., inhibition cannot be overcome by the addition of substrate. They do inhibition by binding irreversibly to the enzyme but not at the active site. Examples include cyanide, which combines with the iron in the enzyme cytochrome oxidase, and certain heavy metals such as Ag or Hg, combine with SH groups. Another example is ethylene diamine tetraacetic acid, EDTA, which blocks all calcium-requiring enzymes because it is a chelating agent and can remove all calcium from the reaction site. Similarly, fluoride inhibits all magnesium-needing enzymes, example enolase, by removing magnesium. The activities of these enzymes reappear on the restoration of the supply of calcium and magnesium ions respectively. Effects of non-competitive inhibition on enzyme kinetics. IKM is unchanged. 2 Vmax is decreased. 3 lines on a double reciprocal plot intersect at the same point on the x-axis. 6.8.1.3 Uncompetitive inhibition. In this case, the inhibitor binds only to ES complex and thus inactivates the enzyme. The reaction is not allowed to go towards the product formation. This type of inhibition usually occurs only in multi-substrate reactions. Examples include siglitazone, used as a therapeutic agent to control various inflammatory diseases, and camptothecin as an anti-cancerous drug. Effects of uncompetitive inhibition on enzyme kinetics. IKM is decreased. 2 Vmax is also decreased. 3 lines on double reciprocal plots are parallel. Figure 6.26 Effects of inhibitors on enzyme kinetics. VO, initial velocity, Vmax, maximum velocity, S, substrate concentration. Figure 6.27 Effects of inhibitors on Lineweaverberg plot. VO, initial velocity, Vmax, maximum velocity, S, substrate concentration, Km, Michaelis-Menten constant. 6.8.2 Irreversible enzyme inhibition. It is a permanent inhibition of an enzyme. It is not reversed by dilution. It is brought about by the formation of a strong covalent linkage between enzyme and inhibitor. The site of the attack is specific amino acid at the active site that participates in an enzymatic reaction. The phenomenon of irreversible inhibition is also known as enzyme poisoning. Irreversible inhibitors form stable covalent bonds with the active sites of an enzyme, usually by the chemical process of alkylation or acylation. There are many naturally occurring and synthetic irreversible inhibitors. These inhibitors are used to identify the amino acid residues at the active site of the enzyme. Incubation of inhibitor with enzyme results in loss of enzyme activity permanently. For example, diisopropyl fluorophosphate, DFP, is an organophosphorus inhibitor used as an insecticide or for enzyme research. DFP can inhibit chymotrypsin by reacting with the active site serine, amino acid number 195, to form DFP chymotrypsin. DFP can also inhibit acetylcholinesterase, a serine protease that hydrolyzes the neurotransmitter acetylcholine. Another example of an irreversible inhibitor is lead, PB. It can permanently block the enzyme ferrochelatase, which is involved in the synthesis of heme. Because of their serious lethal effects, irreversible inhibitors are also known as suicide inhibitors. Figure 6.28 Irreversible inhibition of chymotrypsin at SER 195 by diisopropyl fluorophosphate, DFP. 6.9 Regulation of enzyme activity. All the factors like temperature, pH, substrate availability, zymogens, inhibitors, and the presence of metals and coenzymes help regulate the enzyme activity. Several other factors are also involved in the regulation of enzyme activity. These include synthesis of enzymes, allosteric regulation, covalent modification, endpoint inhibition, feedback inhibition, regulation by modulator protein calmodulin, enzyme/substrate compartmentation, etc. 6.9.1 Regulation of enzyme concentrations. The overall synthesis and degradation of a particular enzyme, also termed its turnover number, is one way of regulating the quantity of an enzyme. The amount of an enzyme in a cell can be increased by increasing its rate of synthesis and decreased by decreasing the rate of its degradation, or both. The induction, i.e., an increase in enzyme synthesis caused by an effector molecule, is a common mechanism. This can manifest itself at the level of gene expression, RNA translation, and post-translational modifications. The actions of many hormones and/or growth factors on cells will ultimately lead to an increase in the expression and translation of new enzymes not present before the signal. The degradation, repression, of enzymes is constantly occurring in the cell. However, the molecular mechanism of enzyme degradation is poorly understood. The turnover number of an enzyme can be used for general comparison with other enzymes or other enzyme systems, yet these numbers can vary from minutes to hours to days for different enzymes. 6.9.2 Regulatory enzymes. These enzymes are concerned with the regulation of various metabolic pathways. Regulatory enzymes are reversibly modulated by effectors. Such enzymes are usually found at the first unique step in a metabolic pathway, the first committed step. Regulation at this step conserves material and energy and prevents the accumulation of intermediates. Two methods of regulation are available: non-covalent allosteric regulation and regulation by covalent modification. 6.9.2.1 Allosteric enzymes. The word allosteric is derived from the Greek allos for other and stereos for shape, or site, meaning other site. These enzymes are specialized regulatory molecules and possess a quaternary structure. They have a second regulatory site, i.e., an allosteric site, different from the active site. Allosteric inhibitors, negative effectors, or activators, positive effectors, bind to this site and regulate enzyme activity via conformational changes in one part of the enzyme that then affect the overall conformation of the active site, causing an increase or decrease in activity. Allosteric effectors or modulators bind non-covalently to the enzymes that they regulate. The state of allosteric enzyme in the presence of an inhibitor is called a tight, T, state, while in the absence of substrate it is called a relaxed, R, state. The addition of substrate increases the concentration of the R state, whereas the addition of inhibitor increases the concentration of the T state. Allosteric inhibition. It is carried out by an allosteric inhibitor or negative effector. Homotropic inhibition, when the inhibitor is the substrate of the same reaction. Heterotropic inhibition, when the inhibitor is the substance other than the substrate of reaction. Feedback inhibition, when the inhibitor is the product of the reaction. Figure 6.29 Feedback inhibition. 2. Allosteric activation. It is carried out by an allosteric activator or positive effector. It may be homotropic or heterotropic. The addition of modulators, effectors, alters the enzyme activity. Activators lower the Km while inhibitors raise the kilometer. Allosteric enzymes do exhibit saturation kinetics at high S, but they have a characteristic sigmoidal saturation curve rather than a hyperbolic curve when VO is plotted versus S. The addition of an allosteric activator tends to shift the curve to a more hyperbolic profile, similar to Michaelis-Menten curves, while an allosteric inhibitor results in more pronounced sigmoidal curves. This sigmoidal shape is supposed to result from the cooperativity of structural changes between enzyme subunits. It is to be noted that a true Km cannot be determined for allosteric enzymes, so a comparative constant like S0.5 or K0.5 is used. Figure 6.30 Effects of allosteric effectors on enzyme kinetics. VO, initial velocity, Vmax, maximum velocity, Km, Michaelis-Menten constant, S, substrate concentration. Example of an allosteric enzyme, phosphofructokinase 1, PFK1, is an allosteric enzyme. It is involved in the regulation of glycolysis. It catalyzes the third step, conversion of fructose 6-phosphate to fructose 1,6-bisphosphate, of glycolysis. Phosphenol pyruvate, PEP, which is an intermediate of the glycolytic pathway, is an allosteric inhibitor of PFK1. ADP is an allosteric activator of PFK1. When the ratio PEP/ADP is high, flux through glycolysis decreases at the PFK1 step. When the ratio of PEP/ADP is low, PFK1 is activated and glycolysis produces more ADP from ADP. Thus, the concentrations of PEP and ADP act allosterically through PFK1 to regulate the activity of the whole glycolytic pathway. Figure 6.31 Reaction catalyzed by PFK1. As ADP is an allosteric activator of PFK1, it lowers the apparent Km without affecting Vmax. For a given fructose 6-phosphate concentration, the velocity of reaction, VO, is larger in the presence of ADP. The allosteric inhibitor PEP raises the apparent Km without changing Vmax. 6.9.3 Regulation by covalent modification. It is another common regulatory mechanism characterized by the reversible covalent modification of an enzyme. The formation of a covalent bond may affect enzyme activity, such as methylation, carboxylation, and phosphorylation. In phosphorylation, a phosphate group is transferred from an activated donor, frequently ADP, to an amino acid on the regulatory enzyme. Usually, this phosphorylation occurs in response to some stimulus, like a hormone or growth factor, that will either activate or inactivate target enzymes via changes in Km or Kat. The phosphorylated form is the active form in some enzymes, while the dephosphorylated form is the active form in other enzymes. Glycogen phosphorylase exists in two forms, an active form that is phosphorylated and another inactive form, which is dephosphorylated state. The CAMP activates CAMP-dependent protein kinase. Protein kinase phosphorylates inactive form of glycogen phosphorylase kinase to active form. The enzyme protein phosphatase removes phosphate and inactivates phosphorylase kinase. On the other hand, glycogen synthase becomes inactive in phosphorylated form and active in dephosphorylated form. Figure 6.32 Regulation of glycogen phosphorylase and glycogen synthase by covalent modification, P, phosphate, Pi, inorganic phosphate, ADP, adenosine triphosphate. Table 6.7 Enzymes regulated by covalent modification via phosphorylation. Enzyme. Active form. Inactive form. Glycogen phosphorylase. Phosphorylated. Dephosphorylated. Citrate lyase. Phosphorylated. Dephosphorylated. Glycogen synthase. Dephosphorylated. Phosphorylated. Pyruvate dehydrogenase complex. Dephosphorylated. Phosphorylated. 6.9.4 Feedback control and inhibition by end product. In the biosynthetic pathway, the end product may directly inhibit the enzyme activity in the early steps. The end product of the enzymatic reaction may control the rate of enzyme synthesis at the level of gene transcription. For example, inhibition of the HMG-CoA reductase enzyme, in cholesterol synthesis, by the level of cholesterol. 6.9.5 Regulation by zymogens. Some enzymes are secreted in inactive forms called proenzymes or zymogens. Activation of proenzymes occurs by removal of the polypeptide chain that masks the active site. Zymogens have the following biological importance. Some enzymes are secreted in zymogen form to protect the tissues of origin from autodigestion. To ensure rapid mobilization of enzyme activity at the time of need in response to physiological demands. Pepsinogen and trypsinogen are well-known examples of zymogens of pepsin and trypsin respectively. 6.9.6 Regulation by modulator protein calmodulin. Many enzymes are dependent on calmodulin and calcium. Calmodulin is a small protein, 17 kDa, that can bind up to 4 calcium ions in the two globular domains. On binding with calcium, calmodulin acts as a protein cofactor to stimulate the activity of target regulatory kinases like phosphorylase kinase, myosin kinase, Ca-ATPase, and a Ca/calmodulin-dependent protein kinase. It is the structural conformation of Ca-calmodulin that makes it an active cofactor. 6.9.7 Enzyme/substrate compartmentation. Isolation of metabolic pathways into distinct subcellular sites like the cytosol or specialized organelles, nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, etc., is another form of regulation. Enzymes related to a given pathway frequently form organized, multi-component macromolecular complexes that perform a particular cellular process. Similarly, it follows that the substrates associated with a given pathway can also be localized to the same organelle or cytosolic location. This separation allows for more specialized regulation of cellular processes. For example, fatty acids are synthesized by one set of enzymes in the cytosol endoplasmic reticulum and the Golgi apparatus and used by a different set of enzymes as a source of energy in the mitochondria through beta oxidation. Summary: enzymes are biocatalysts, organic substances, mostly protein in nature, heat label and synthesized by living cells. Enzymes speed up chemical reactions several folds, 103 to 108 times faster than non-enzymatic reactions. All enzymes are classified into 6 major classes according to the reaction being catalyzed. Oxidoreductases conduct redox reactions. Hydrolases catalyze hydrolytic cleavage of CC, CO, CN and other covalent bonds. Liases remove or add H2O, NH3, or CO2 molecules into the substrate. Ligases, synthetases, catalyze ligation or joining of two substrates in various reactions of synthesis. Coenzymes are small organic molecules, that may be co-substrate, NAD+, or prosthetic group, FAD. Cofactors, these are the non-protein part of complex enzymes in the form of metal ions such as Mg2+, Ca2+, Zn2+, Fe2+, etc. The active site is a small pocket-like part of the enzyme, responsible for catalytic activity. Enzymes are highly specific in their actions, i.e., one enzyme can act on one specific substrate only. Newly synthesized enzymes are called proenzymes or zymogens, they are inactive. Isoenzymes/isozymes are the enzymes of the same organism performing the same catalytic activity but physically and chemically distinct. The energy of activation, EAct, is the minimum amount of energy required by the substrate to become activated complex or the transition state. The nature of binding between enzyme and substrate may be lock and key or induced fit. Enzyme kinetics is the study of the rates of chemical reactions that are catalyzed by enzymes. The activity of the enzyme is affected by many factors like concentration of enzyme, the concentration of substrate, temperature, pH, presence of metal ions, coenzymes, and inhibitors. Michaelis-Menten constant, Km, is the substrate concentration at which the rate of reaction is 1/2 Vmax. Optimum temperature is the temperature at which the activity of an enzyme is best. The optimum pH of pepsin is 1.5 to 2.0, for amylase, it is 6.4 to 6.9 and for alkaline phosphatase, it is about 9.5. Inhibitors are chemicals that reduce the rate of enzyme-catalyzed reactions. In classical competitive inhibition, there is a great structural similarity between the substrate, S, and inhibitor, I. S and I can bind to the same site on enzyme, E. Irreversible enzyme inhibition is the permanent inhibition of an enzyme. It is not reversed by dilution. It is brought about by the formation of a strong covalent linkage between enzyme and inhibitor. Allosteric enzymes are specialized regulatory molecules having a quaternary structure and possess a second regulatory site. Enzymes are regulated by covalent modifications such as methylation, carboxylation, and phosphorylation. References. Exercise questions. 4 MCQ 1 to 3C Part 1 of 2. MCQ 4. Michaelis-Menten constant, Km, is the substrate concentration at which the rate of reaction has which of the following relation with maximum velocity, Vmax? Double of Vmax. Equal to Vmax. Half of Vmax. 1/4 of Vmax. 1/3 of Vmax. MCQ 5. A 45-year-old man suffering from lobar pneumonia is on treatment with the sulfonamide group of antibiotics. Which of the following enzymes is inhibited by the drug used by this patient? PABA decarboxylase, PABA dehydrogenase, PABA lyase, PABA oxidase, PABA synthetase. MCQ 6. A 22-year-old man in a semi-conscious state was rushed to the emergency with vomiting, sweating and decreased heart rate. History revealed that the patient developed symptoms after spraying pesticide in the cornfield. This condition of the patient was expected due to inhibition of acetylcholinesterase by covalently binding organophosphate to it. What type of inhibition is this? Classical competitive inhibition. Irreversible inhibition. Non-classical competitive inhibition. Non-competitive inhibition. Uncompetitive inhibition. Please write answers in the comments. End of Part 2 of 2 of Chapter 6. Thank you.